Prokaryotic gene regulation. The DNA of prokaryotes is organized into a circular chromosome supercoiled in the nucleoid region of the cell cytoplasm. Proteins that are needed for a specific function, or that are involved in the same biochemical pathway, are encoded together in blocks called operons. For example, all of the genes needed to use lactose as an energy source are coded next to each other in the lactose, or lac, operon. In prokaryotic cells, there are three types of regulatory molecules that can affect the expression of operons: repressors, activators, and inducers. Repressors are proteins that suppress transcription of a gene in response to an external stimulus, whereas activators are proteins that increase the transcription of a gene in response to an external stimulus. Finally, inducers are small molecules that either activate or repress transcription depending on the needs of the cell and the availability of substrate. The TRP operon, a repressor operon. Bacteria such as E. coli need amino acids to survive. Tryptophan is one such amino acid that E. coli can ingest from the environment. E. coli can also synthesize tryptophan using enzymes that are encoded by 5 genes. These 5 genes are next to each other in what is called the tryptophan TRP operon. Figure 1. If tryptophan is present in the environment, then E. coli does not need to synthesize it, and the switch controlling the activation of the genes in the TRP operon is switched off. However, when tryptophan availability is low, the switch controlling the operon is turned on. Transcription is initiated, the genes are expressed, and tryptophan is synthesized. A DNA sequence that codes for proteins is referred to as the coding region. The 5 coding regions for the tryptophan biosynthesis enzymes are arranged sequentially on the chromosome in the operon. Just before the coding region is the transcriptional start site. This is the region of DNA to which RNA polymerase binds to initiate transcription. The promoter sequence is upstream of the transcriptional start site. Each operon has a sequence within or near the promoter to which proteins, activators or repressors, can bind and regulate transcription. A DNA sequence called the operator sequence is encoded between the promoter region and the first TRP coding gene. This operator contains the DNA code to which the repressor protein can bind. When tryptophan is present in the cell, 2 tryptophan molecules bind to the TRP repressor, which changes shape to bind to the TRP operator. Binding of the tryptophan repressor complex at the operator physically prevents the RNA polymerase from binding and transcribing the downstream genes. When tryptophan is not present in the cell, the repressor by itself does not bind to the operator. Therefore, the operon is active and tryptophan is synthesized. Because the repressor protein actively binds to the operator to keep the genes turned off, the TRP operon is negatively regulated and the proteins that bind to the operator to silence TRP expression are negative regulators. Catabolite activator protein, CAP, an activator regulator. Just as the TRP operon is negatively regulated by tryptophan molecules, there are proteins that bind to the operator sequences that act as a positive regulator to turn genes on and activate them. For example, when glucose is scarce, E. coli bacteria can turn to other sugar sources for fuel. To do this, new genes to process these alternate genes must be transcribed. When glucose levels drop, cyclic AMP, CAMP, begins to accumulate in the cell. The CAMP molecule is a signaling molecule that is involved in glucose and energy metabolism in E. coli. When glucose levels decline in the cell, accumulating CAMP binds to the positive regulator, catabolite activator protein, CAP. a protein that binds to the promoters of operons that control the processing of alternative sugars. When CAMP binds to CAP, the complex binds to the promoter region of the genes that are needed to use the alternate sugar. Sources. Figure 2. In these operons, a CAP binding site is located upstream of the RNA polymerase binding site in the promoter. This increases the binding ability of RNA polymerase to the promoter region and the transcription of the genes. The LAC operon, an inducer operon. The third type of gene regulation in prokaryotic cells occurs through inducible operons, which have proteins that bind to activate or repress transcription depending on the local environment and the needs of the cell. The LAC operon is a typical inducible operon. As mentioned previously, E. coli is able to use other sugars as energy sources when glucose concentrations are low. To do so, the CAMP-CAP protein complex serves as a positive regulator to induce transcription. One such sugar source is lactose. The LAC operon encodes the genes necessary to acquire and process the lactose from the local environment. CAP binds to the operator sequence upstream of the promoter that initiates transcription of the LAC operon. However, for the LAC operon to be activated, two conditions must be met. First, the level of glucose must be very low or non-existent. Second, lactose must be present. Only when glucose is absent and lactose is present will the LAC operon be transcribed. Figure 3. This makes sense for the cell, because it would be energetically wasteful to create the proteins to process lactose if glucose was plentiful or lactose was not available. If glucose is absent, then CAP can bind to the operator sequence to activate transcription. If lactose is absent, then the repressor binds to the operator to prevent transcription. If either of these requirements is met, then transcription remains off. Only when both conditions are satisfied is the LAC operon transcribed. Section summary. The regulation of gene expression in prokaryotic cells occurs at the transcriptional level. There are three ways to control the transcription of an operon: repressive control, activator control, and inducible control. Repressive control, typified by the TRP operon, uses proteins bound to the operator sequence to physically prevent the binding of RNA polymerase and the activation of transcription. Therefore, if tryptophan is not needed, the repressor is bound to the operator and transcription remains off. Activator control, typified by the action of CAP, increases the binding ability of RNA polymerase to the promoter when CAP is bound. In this case, low levels of glucose result in the binding of CAMP to CAP. CAP then binds the promoter, which allows RNA polymerase to bind to the promoter better. In the last example, the LAC operon-two conditions must be met to initiate transcription. Glucose must not be present, and lactose must be available for the LAC operon to be transcribed. If glucose is absent, CAP binds to the operator. If lactose is present, the repressor protein does not bind to its operator. Only when both conditions are met will RNA polymerase bind to the promoter to induce transcription. Thank you for watching BookTube channel. Make sure to smash the like and subscribe buttons to receive notifications about new contents.